Welcome everybody to yet another Journal Club. February 2021. I keep thinking it's still 2020. It's just like the year's never ended almost. It just keeps going on and on, doesn't it? So a blast from the past in I guess celebration of the never-ending 2020. We have a paper from 2020. I actually thought this was a more recent paper, but it's from February, I believe, of 2020. Genes and development paper. Pretty meaty paper. So welcome to our Journal Club. I'm Oliver Medvedik. This is presenting to you from Life Extension Advocacy Foundation, as always, and broadcasting from a slightly different location in New York City. So let's discuss the paper that we're going to pull up today. It's a mitochondrial signaling paper. It's a little bit different. It's I think we've focused on a number of papers that we were doing a lot of papers that focused on clinical trials, and we're back to the science here, but the paper does end with a potential for clinical trials. So let me share my screen. Hopefully the screen sharing is working pretty well. There we go. Hopefully that looks okay. Okay. So the paper title is Mitochondria and Nucleus Retrograde Signaling Drives Formation of Cytoplasmic Chromatin in Inflammation and Senescence. A pretty big title there. A lot of stuff taking place by Visioli et al., number of labs, Harvard Stem Cell Institute, Cancer Research, UK Beatson Institute in Glasgow, et cetera, et cetera. UPenn, University of Pennsylvania School of Medicine. So quite a lot of work. So there's about 7 figures here. I'm not sure if we're going to get through all of it, and each figure obviously has a bunch of subfigures. So what is this all about? Well, what are the authors are trying to, what's their kind of main message here? Well, we all talked about hallmarks of aging. So we all know that there's several key hallmarks of aging that were published a number of years back, I believe in 2013, where number of these factors such as senescence, mitochondrial dysfunction, insulin signaling pathways that all basically converge on aging. And what this paper seeks to do, among other things, is to show that some of these disparate hallmarks are actually connected, particularly mitochondrial dysfunction, also senescence, and also inflammation, which is another hallmark. So inflammaging is the term that's usually used in association with senescence-associated secretory proteins, CCFs, that are released by senescent cells. So there's a connection being made between these several different hallmarks of aging in this paper. And an interesting component of all of this, all of these interactions is one of the PIs in this lab, what they study is kind of the byproduct of this is something called CCFs, which are cytoplasmic chromatin fragments. So essentially the way I would summarize this is that you have bad mitochondria that are dysfunctioning, they're not being repaired, there's no mitophagy taking place or insufficient, which means that the mitochondria are cleared out. And that signals the nucleus somehow through retrograde signaling, meaning normally you have the nucleus produce mitochondrial proteins which get incorporated into the mitochondria. So you have a flow of information from nucleus to mitochondria, but in this case you have mitochondria signaling back to the nucleus, and then bad things happen. And one of the bad consequences are that you have these things called, they abbreviate CCFs, or cytoplasmic chromatin fragments. So you have this blebbing. So from the nucleus, you get sort of these chromatin fragments. They get released into the cytoplasm, and those trigger the senescence associated secretory phenotype, SASP, which are basically inflammatory markers. And then things kind of go haywire from there. The cells enter senescence, they trigger inflammation around themselves, and so on and so forth. So that's the connection that they're making in this paper, and they're trying to put in the pieces as to how the signaling happens between the bad mitochondria, the nucleus, and then the release of these CCFs, or chromatin fragments. So a lot of these pieces weren't known until this paper was published. And jumping ahead to the end here, one takeaway point from this paper is that at least the release of these CCFs and the attenuation of, ameliorating the release of the CCFs and then the subsequent attenuation of SASP can be promoted by HDAC inhibitors, so histone deacetylase inhibitors. So, and I believe in the paper they use trichostatin A and another one. So, having some genes turned on by inhibiting HDAC, so HDACs normally silence genes. So turning on some genes that are normally turned off through this retrograde signaling from bad mitochondria in the nucleus, somehow we'll see how it blocks the formation of these CCFs. So that's a particular, that's a potential therapeutic target, is giving low doses of HDAC inhibitors. And I'm going to throw this out there as well, and we'll, they mentioned this in the paper, but they didn't try to figure it, is that this is all dose dependent. If you give, too much HDAC inhibitors, you can promote senolysis, clearing of senescent cells. But if you give a lower dose, you can basically ameliorate some of these effects without having a senolytic response. So that's the, I'm going to stop sharing. So that's the kind of gist of the paper, and we'll kind of take it from figure to figure and show some of the evidence that they present. One thing that I'd like to mention here, because there's a lot of moving parts, so maybe some of this stuff is still kind of, still sort of, the pieces are, you're still trying to put together in your head. Going back, way back to the 1990s, this kind of reminds me, and I haven't read any of the past papers that are connected to this, but CCFs, cytoplasmic chromatin fragments, that reminds me a lot about ERCs, or extrachromosomal rDNA circles in yeast. I don't know if anybody remembers this, but this was work that was done at the Sinclair lab way back in the 1990s. Yeah, we're going over a quarter of a century ago there, Steve. That is.
Oh, thanks for that. Thanks for that.
Yeah, so, payphones were still used unironically, just letting you know. So yeah, so yeast, for example, which is a model organism that I use in my studies a lot in the past, they senesce, they age. There's a number of different ways that they do this, and one mechanism that was studied at the Parenti lab at MIT at the time, which David Sinclair was also part of these studies, along with I believe Brian Kennedy, Matt Haberlin, and others, was the formation of these ERCs, which were basically bits of the ribosomal locus would be copied out and would accumulate. And it was shown that they would be toxic. So you would get this this accumulation of chromatin material, and I'm not really sure if it actually, I don't remember if it leaked out into the cytoplasm, but basically it was chromatin instability. And this kind of reminds me of this because they're looking at mammalian cells here, and you get these cytoplasmic chromatin fragments that leave the nucleus and bleb out. So I don't know how much of a mechanistic connection there is, and I'm not 100% sure as to what chromatin locus these cytoplasmic chromatin fragments are derived from specifically. It wasn't mentioned in this paper, but no doubt it was mentioned in prior papers. So I'm just throwing that out there as a species, across many genera, connection that may have some evolutionary significance. I don't know. Just to remind everybody that it might not be that species specific, this type of genomic instability that arises. But anyway, this triggers SASP, these CCFs, and they were interested in this paper to figure out what triggers the formation of these CCFs as far as signaling is concerned. So before we go in there into this paper, I'm just going to jump ahead a little bit and say that one of the things that was noticed in this paper, and this is the last comment I make, kind of keep us on track, is that the signaling from bad mitochondria that have basically problems with their membrane polarization, so on and so forth, those signals are reflected in the nucleus through downregulation of genes that are required for oxidative phosphorylation in mitochondria. So it's kind of squelching those genes and not allowing, mitochondria to be, I guess, sufficiently repaired. And one of the hunches is that HDAC inhibitors get those genes to be re-expressed again, right? Anyway, so let's go back to this paper here. Share my screen. Okay. I don't know if anybody had a chance to read this paper here. There's a lot of jargon here. Okay. And we'll scroll until we get to figure 1. And here's the figure. So one thing that they establish is that mitochondria are required for the formation of the CCFs, which are cytoplasmic chromatin fragments. And what data do they use? So they do RNA-seq analysis. So IR, so proliferate, PROF is just regular proliferating. I think IMR 90 cells is that they use through, I forgot which, what mammalian cell was IMR 90? Anybody know off the top of their head?
They're lung fibroblasts.
Lung fibroblasts. Okay, so they use lung fibroblasts throughout the paper primarily, except until they get to the end where they start doing a mouse model in vivo. So figure 7 is when they switch to in vivo. Everything up to this point from figure 1 through 6, I believe, is in vitro, primarily IMR 90 cells. So they do RNA-seq. They show PROF proliferating cells, and then IR is ionizing radiation. So they basically induce damage in a number of ways here. They induce mitochondrial damage by using this. So first of all, ionizing radiation. So what do they see? They see basically a fold change in a number of RNA transcripts that correlate to a lot of different things. So we have a downregulation of nuclear encoded mitochondrial genes, so complexes 1, 2, 3, 4, 5, after basically ionizing radiation treatment over a number of days. They also, what they do is they use this decoupling factor called CCCP, which basically messes up mitochondria. So that's, and what they do then is they clear, so they basically also then clear the mitochondria using Parkin, which is basically a means to target the mitochondria using, blanking here, ubiquitin ligase, or basically target them for mitophagy. So let's see, figure ECD, what do they look at? So proliferating ionizing radiation. So as a consequence, they get superoxide dismutase levels go up, so messenger RNA levels go up. Mitochondrial reactive oxygen species go up, and MMP is, keep going here, it's too much jargon. Mitochondrial membrane potential goes down. So basically, the mitochondria gets screwed up in response to ionizing radiation. Okay, so going to figure E, F, and G. So basically, what they do here is, again, what they're trying to correlate here is they're going to show later. They're basically establishing the fact that mitochondria are getting screwed up in the context of ionizing radiation, and that some of these effects that they see later, particularly these CCFs, cytoplasmic chromatin fragments, are due to the presence of mitochondria, that there's going to be mitochondrial signaling involved, and that if they clear out the mitochondria, that this goes away. So it's just a lot of all of this stuff leading up to that. So, Parkin is basically, they express this ubiquitin ligase that basically targets mitochondria for mitophagy, after they add this decoupling factor CCCP, which basically messes up the membrane potential of mitochondria, and then they clear out. And this in figure F and G, they have this TOM20 factor, which is a mitochondrial component. It's basically, I believe it's a membrane protein that's involved in transporting proteins. So it's a mitochondrial factor. So basically it stains mitochondria. Okay. So what you could see here is they're establishing that if they treat these cells with this with Parkin, after they've induced with ionizing radiation and also this decoupling factor, you don't have any TOM20 present, so the mitochondria go away. I don't know how long the cells could live without mitochondria. So you basically have mitochondrial less cells. So that kind of gets to figure H, which basically shows that gamma H2AX is a histone that's associated with DNA damage, and it could be found in the nucleus, but also out of the nucleus. And so it associates with the CCFs. So you notice here that the blue is in DAPI in figure H. Proliferating cells, DAPI, H2AX, nothing, merge, blue. Parkin expression, so ionizing radiation, and then CCCP, which is basically decoupling specifically mitochondrial membrane potential. So you have no mitochondria evidently in this IR-CCCP spot with the ionizing radiation component you do. So if you have if you do have mitochondria present, you still have the presence of these cytoplasmic chromatin fragments, which are denoted in this green. But if you get rid of the mitochondria, they go away. They're drastically attenuated. So, so they're suggesting here that you have no mitochondria and you basically are attenuating or removing getting rid of these fragments, these cytoplasmic fragments. And as a consequence, you also, when you get rid of the mitochondria, you have a number of these inflammatory markers such as IL-1A alpha, IL-1 beta, IL-6, IL-8 also get attenuated considerably. So messenger RNA levels of those go down if you don't have mitochondria present. And they do that across the board for a number of other factors. So J is, where's J? Oops, I can't move this. Whoops. Where is J? Not H. I can't see J. Okay, I can't see J. Okay. Well, you can sort of zoom in on a number of factors here. So basically by getting rid of the mitochondria, you could see controlled proliferation, Parkin proliferation. So looking at Parkin IR CCCP, you see that basically the levels go back to, well, not quite normal, but you don't have a number of these senescence associated factors. Basically, the numbers go back to more looking like normal than if mitochondria were present. So I'm going to stop sharing here. That's a lot of stuff. That's just figure one. So basically, they are trying to establish here that mitochondria, or presence of mitochondria, damaged mitochondria specifically, is perhaps one of the driving forces for the formation of these cytoplasmic chromatin factors, along with a number of other senescence-related factors, such as the various proteins that were shown there in in messenger RNA form, that may be connected to the two, but they haven't made that connection yet. So, any questions there so far? So, it's a lot of stuff.
I just want to mention that mitochondrial ablation experiment was bizarre to me. I've never seen anything like that.
Yeah, I've never done that, so I have to take their word for it. I mean,
I was kind of skeptical at first of their claim that they actually did it. They ablated everything, but panel D was pretty persuasive, and it was like, I think 6 to 10 days post treatment that they actually get the cells like that. So kind of a weird treatment. It's kind of like removing the whole mitochondria is like removing the whole a liver from a person and saying, you need a liver to survive.
Yeah. I mean, yeah. I mean, I guess my 30,000-foot altitude viewpoint on this is that the cell. I'm like, wow, the cells are still alive. And number two, okay, the chromatin. I guess it's impressive that the chromatin, the cytoplasmic chromatin fragments go away, and nothing else seems to go away, or kind of go explode. I mean, they haven't looked at it. I mean, I don't looked at a bunch of other things. I mean, I don't know. They go away. So it's. You would think getting rid of mitochondria, maybe other crazy things would happen, like maybe you would get more CCFs, but no, those CCFs also go away, which, okay, that's good direction, I guess.
Yeah, I'm guessing it's not a specific intervention. I'm guessing a whole lot of stuff happens.
Yeah. I mean, and then they try to put the pieces later in to basically show that, like, okay, well, what's going on is there's a June kinase signaling pathway that they invoke later, and then they basically try to put pieces from mitochondria. I mean, since we're talking about this here, it does seem rather drastic. I would think maybe there's some other more subtle ways that you can inhibit retrograde signaling without basically just wiping out the entire health of mitochondria. But I guess getting this to work would strongly be suggested that then more subtle approaches would be effective, right? Just getting rid of the mitochondria altogether gets you clean data that shows that CCFs go away, then basically blocking out other things will also work. I think they do that. It's just I'm having memory issues here because there's like 7 figures and like just cram all that into my head, and I'm pulling that all back out again. And there's just a lot of acronyms flying around here. It's the first time I realized that there was another second messenger molecule. It's actually a cyclic GMP AMP that's coupled. Did you know that?
Not familiar, no.
Yeah. So there's obviously there's calcium, there's cyclic AMP, there's the inoxidyl phospholipids, but they introduced this one second messenger that I had to look up. I was like, wait, cyclic GMP AMP, like sort of a circular link of adenosine monophosphate and adenosine monophosphate. It turns out it was discovered like less than 10 years ago. I was like, well, all right. So anyway,
cool.
Yeah, it's just more things for me to process while I'm trying to get the data here as well. Okay. So back to the paper. So so they ablate the mitochondria and CCFs go away. So let's see. Let me just pull up CCCP here. Yeah, second messenger cyclic GMP AMP, CGAMP, which basically is involved in signaling from which they'll invoke later, which is in the damage response pathway. Importantly, we discovered that CCF formation depends on a nuclear lamin B interaction and sensors. CGAS, which produces the second messenger cyclic GMP AMP. So I did not know that. So there you go. Okay, where's I just wanted to pull up the CCCP just to make sure that I have that, know exactly what they're doing to ablate this. I know it's supposed to decouple the membrane potential of mitochondria. I don't know exactly how it does it. 2 days after irradiation, cells were treated with mitochondrial uncoupler carbonyl cyanide M chlorophenyl hydrazone, CCCP, to induce mitochondrial depolarization, and then Parkin-mediated mitophagy. So yeah, ionizing radiation will induce a damage response that's more generic, obviously double-stranded breaks in DNA, etc. Various oxidizing species. And then this mitochondrial uncoupler will target mitochondria more specifically. And we confirmed that expression of mitochondrial proteins was dramatically reduced in Parkin CCP-treated senescent cells, but not in control CCP-treated senescent cells as assessed by immunoblotting, 1F. So immunoblotting is, whoops. So scroll down here. Yeah. So that was here. Okay, so these are also it was a downregulation of nuclear encoded. So yeah, one thing I think I omitted from figure 1, just to squeeze out some more information. So yeah, so not only you get rid of the mitochondria, you also you also downregulate genes that are involved in mitochondria that are basically incorporated in mitochondria, so proteins. So that's figure F. So maybe not that much of a surprise. The cell senses there's no mitochondria, so it downregulates the proteins. So let's go on to figure 2. Big meaty figure. So here they look at reactive oxygen species, and they basically implicate ROS in the formation of these CCFs and also the expression of SASP. So we already saw that there was senescence-associated secret phenotype markers that are associated when you basically ablate and get rid of the mitochondria. We're actually, no, I think they're blocked if you ablate the mitochondria, sorry, using Parkin, but if the mitochondria are damaged, they go up. So implicating in a very, very crude way that there's mitochondria involvement. So now they're basically looking at the role of specifically oxidation and looking at reactive oxygen species. I'm trying to basically get my head around as to kind of what the main takeaway point of this is. Just gonna look at the figure here. So again, ionizing radiation is IR. CCCP is basically this uncoupling factor that basically messes up mitochondria. So these are positive cells for mitochondrial reactive oxygen species, so MROS. Yeah, mitochondrial reactive oxygen species measured by flow cytometry using mitostocks in proliferating incessant cells of control and Parkin expressing cells with or without CCP treatment 10 days after ionizing radiation. So, so when they treat, so clearly when they treat the cells with Parkin, so Parkin is basically ablating. So when you see this Parkin, it's they're getting rid of the mitochondria. Right? It's mitophage is taking place. So so I don't know. It just seems that yeah, mitochondrial obviously reactive oxygen species go away if you don't have mitochondria. So I'm not entirely sure what this is supposed to really demonstrate here, to be honest, because you're adding in H2O2 hydrogen peroxide and you have MROS go up, which I would think is not too surprising, that you would basically stress the system and you have this go up. What they do show here, though, that if you have also, I believe if you have reactive oxygen species go up, you also get cells enter senescence. So later they're going to tease out kind of two different senescence responses, which is cells entering cell cycle arrest, which is obviously dependent on tumor suppressors like P21. So if you have stress coming in from a reactive oxygen species, the cells are also entering senescence. They're basically halting their cell cycle. They're also releasing, they're also entering a SASP phenotype, and you have the formation of these CCFs. And I think what later they're going to show is that they can kind of tease apart some of these factors. So they'll show that you don't necessarily get the cells enter cell cycle arrest by just ablating mitochondria, which is a little surprising because it's a very energetic process. Okay. So what are we looking at here? So yeah, so if you decouple, so basically you mess up the mitochondria, you get much more reactive oxygen species being produced. So that goes up. And see what else, what are the take-home messages here? I think the important part is to look at what's taking place in context of the cytoplasmic chromatin fragments here, where we actually now try to squelch the reactive oxygen species by adding in this compound called MitoPQ. So let me go back to this here. Treated with MitoPQ. This is an antioxidant, I believe. Is anybody familiar with MitoPQ? MitoQ. I think there's a variant of it that's used as a supplement.
Yep.
And it somehow gets into the mitochondria and sops up. I don't know. Does it sop up free radicals, H2O2? I'm not exactly 100% sure.
I think it's an antioxidant, yeah. Although that word gets overused quite a lot. But yeah, I think it is a mitochondrial antioxidant. Michael, I said Todd in the bloody chat. It's Michael on the International Space Station as a question. I think it's that, or it's a TIE fighter. Is it a TIE fighter, or is it the International Space Station?
No, that's the ISS.
I think it would be pointed the opposite way. That would be the viewport of the TIE fighter.
It also looks a bit like, I keep thinking it could also be the Death Star from Empire Strikes Back, where the Emperor sits on his throne and he's got that round window behind him. I wonder if it opens.
Maybe they modeled the ISS after that scene.
Yeah, it's entirely possible.
Regarding figure 2, I think the context here is they're just teeing up for their big figure, which I think is figure 3. Like a lot of the results they have here, they already kind of published in their first CCF paper, the 2013 one. Okay. That paper was like already tying senescence to CCF, but the big difference was they were looking at like replicative senescence. Whereas in this one, they're doing hydrogen peroxide. So like nowadays, a lot of the time is if you want to artificially induce senescence in vitro, you use hydrogen peroxide right at around the concentration they're using it now. So my read is this is just more of a aging relevant senescence inducer than what they'd used in previous papers. But yeah, it's not anything, I don't think it's anything very new.
Okay, all right. So I guess maybe skipping to H and K, which they basically show that adding in this MitoPQ, which is basically suppressing various reactive oxidative species. Well, wait a minute. Why would you get more positive cells? MitoPQ. No. Wait. MitoPQ is why would you get more MROS if you get rid of? Maybe there's MitoQ and MitoPQ. Maybe MitoPQ is an opposite one. Hold on a second. I know what MitoQ is. Proliferating cells of MitoPQ. I have to name these things so similar. God, where's the definition of MitoPQ?
It looks like MitoPQ is like a paraquat derivative, so it's a really nasty.
Okay, all right. So MitoQ good, MitoPQ bad. Okay, so MitoPQ is a paraquat, so that's actually produced. So that's why I was looking. I was like, why does reactive oxygen species go up when you add MitoPQ? Okay, so it's a paraquat derivative, so it's actually driving the formation of this of these oxidative species. Okay, well that makes sense. All right. So okay, so you get more chromatin. So more oxidative stress, more chromatin, cytoplasmic chromatin fragments, and that is reflected in figure K as well. So, so I think I'm just going to leave it at that. The one thing that they do show is that it doesn't seem to affect or actually ionizing MitoQ. So getting rid of, so MitoQ is getting rid of this oxidative response, it doesn't seem to affect cell cycle arrest. So actually, so they're adding in MitoQ here and EDU, which is a marker for replicating cells. So adding in an antioxidant doesn't seem to. So if you add in this MitoQ, not MitoPQ, MitoQ, you basically attenuate your mitochondrial reactive oxidant species. The CCF goes down as well, which is the cytoplasmic chromatin fragments. And you also get an attenuation of factors in the senescence associated secretory phenotype, but the cells are still not exiting cell cycle arrest. So they're kind of separating these functions out here, showing that you get oxidation go up because of defective mitochondria, you get SASP, you get CCFs. You can ameliorate those by getting rid of this oxidative stress. But the other additional thing that that happens in these cells that are senescent, which is they're in cell cycle arrest through messed up mitochondria, is not due to increased oxidative stress from the mitochondria. So if that makes sense, I think. So basically, they're trying to show that this part of the signaling from the mitochondria, it's part of it is due to this oxidative stress, but other parts of it are that affect downstream parts of senescence, such as cell cycle arrest, aren't due to the oxidative stress. Which there was a figure that basically showed with arrows where all of this is actually pointing in different directions, because you have a smorgasbord of figures here. It's like one giant picture would be nice, which would show where all these figures tie into just a nice hypothesis of where all of this is flowing. I have to just sketch it out in my notebook. There's a lot of arrows going back and forth inside of nucleus, outside of nucleus. Okay. All right. So in figure 3 is where they basically try to tie in some signaling as to, okay, so you've got mitochondria, they're defective, you're basically leading to the formation of these cytoplasmic chromatin fragments. What is actually responsible for this activity? And I believe one thing they invoke here is this June kinase signaling, which is a signaling pathway that's implicated in many kind of signaling events. And this signaling pathway is upstream of the formation of CCFs, right? So somehow the signals get from bad mitochondria to the nucleus, and then you get this J and K signaling happen. So here we take a look at figure A, where you have proliferating cells, IMR90, that haven't been hit with ionizing radiation, and then IR 82510. And you see phosphorylation of J and K, which means that the signaling pathway is being activated. And then they have also here as a control, they have this this June kinase inhibitor. I don't know what they use as an inhibitor. I have to scroll down here. With or without J and K. Somewhere in the materials and methods, it says what the inhibitor is, some small molecule probably. And so basically what they're showing here is that this signaling pathway, so at least this signaling pathway here, looking at a Western blot here, I believe. So this is phosphorylated retinoblastoma protein, cyclin A, P21. So what are we looking at here? So yeah, we'll get back to that later. I'm going to jump to figure C, which is basically they're looking at now. Well, back to the just to center ourselves back to cytoplasmic chromatin fragments. Is this kinase, this June kinase, implicated? I guess the short answer is probably yes. And here's basically cross-sectional staining. So gamma H2AX, which is a histone marker that's basically found also in these blebbed cytoplasmic chromatin fragments, which are outside. And if you add in this Jun kinase inhibitor, they get really radically attenuated. So basically you get H2AX ends up in the nucleus, but it's not exported out. So you have this decrease. Yeah, question. So yeah, so I can't recall why they got on the they mentioned it in the introduction, why they thought that Jun kinase signaling was implicated, and I believe it was upstream of this factor called 5,3-BP1, which they implicate in which is used, I believe, I'm trying to, without scrolling back and forth, I think it's involved in resection of DNA that's been cut, and that leads to CCF formation. And Jun kinase signaling, I think, somehow is upstream of 5,3-BP1, which is then implicated. So sometime in the past they implicated 5,3-BP1 in CCF formation potentially, and it's potentially that's how they started to look at Jun kinase. That's upstream of that. So no doubt it's explained a little bit better than that somewhere. So that's why they started to look at this Jun kinase. And it seems that if they attenuate it by using a Jun kinase inhibitor, then CCF levels go down. And of course, when they also throw on this inhibitor, a bunch of senescence-associated secretory phenotype factors like IL-1 alpha, IL-1 beta, IL-8, which are implicated inflammation, the messenger RNA levels also go down. And this is basically a PCA plot, which basically shows that basically a clustering of kind of the expression, and shows you what the similarity is when you add in the Jun kinase inhibitor versus ionizing radiation. It basically shifts to a more normal state. So basically, all these figures here, without belaboring the point, basically show that adding in a Jun kinase inhibitor, and what else do they do here? They add in a short hairpin turn RNA to basically also, so what is this? They're also knocking out, yeah, so they're using a different approach to knock out Jun kinase, I believe, here. So this is a control, and CCF, cytoplasmic chromatin factors, go up in a control, and then they go down when you knock down Jun kinase. So basically, they're implicating Jun kinase, which, as the first sentence says, J&K mediates formation of CCFs and expression of SASP. Right. So they just put that into the puzzle here to build this model up. I'm surprised they don't have a figure that just shows all this stuff. Kind of nice. So I'll just basically go zoom into the figure and stare at it, so I don't get lost in the woods here. But no, I've got to keep this all in my head. All right, so figure 4. So here is when they look at this factor called 5,3-BP1 that I mentioned earlier, which I believe is downstream of Jun kinase signaling, which actually is implicated in, make sure I got this right, I said DNA resection of DNA that has a double-stranded break, but
yeah, it is. 53BP1 is one of the big players in the double-strand break repair pathway. It like brings the whole story together, and it's probably like how you even get the chromatin fragments in the first place, because if you have like unrepaired double-strand breaks, well, there's a fragment which you can potentially bleb off.
Good. All right. So I'm not too far off track here. So good. So it's a central player. So this is really getting like you said, Michael, really to the kind of the heart of this is where it starts. This is where the CCFs basically start. So we've meandered our way to figure 4 from figure 1, from the mitochondria to the nucleus to double-stranded breaks, and we are at the double-stranded breaks where they're actually forming and potentially leading to the formation of these blebbed CCFs, which then lead to a whole bunch of downstream things like SASP and other stuff. So what do they show here? Ionizing radiation, DAPI, you got blebs, gamma H2AX found inside and outside. So it's basically implicated with damaged DNA, but it's also found in heterochromatin, and it's also found in these blebs, which are released. I'm not sure what the blebs are actually. I haven't read the earlier paper, so I don't know what they're. So just as a side here, maybe Michael, maybe somebody else. Besides, is there any? What are these blebs doing? Is this just a way for the cell to get rid of damage and get it basically secreted out of the cell, or maybe thrown into the lysosomes, or does it just accumulate, or what do they? Or is it just toxic, just floating around?
Yeah, I'd love to know the answer to that. This lab seems to be one of the few players who's actually like looking into this cytoplasmic chromatin. They keep showing gamma H2AX, which is like one of the best markers for DNA damage. So when they show the blebs that are like bright green in panel A, it kind of suggests that that's like unrepairable stuff, or like maybe it's double-stranded break repair discard that's just going into the lysosome because you don't need it.
Yeah.
But they haven't sequenced any of this stuff. There's still some H2AX in the nucleus. Like, I don't really know. I also kind of want to know what's left over. Like, if you just snipped out a chunk of chromosome 5, is there a duplicate has there been a successful repair in the nucleus? Or are you just missing a gene now?
Yeah, well, one thing that they mentioned later in the paper, which I found interesting, I like how certain pieces of the puzzle flow together, which is like, and I know this, supposedly if you put DNA into a cell through whatever electroporation or however you do it into the cytoplasm, you trigger like stress responses like SASP that are basically the cell will maybe related to this kind of CCF. Although I think the CCF stuff is wrapped around in a membrane, because it says it's blebbed out of a nucleus. It's not just freely floating DNA. It's wrapped up somehow, probably in a double membrane. I don't know. So I don't know. I find this intriguing, right? Because it reminds me of what's going on in yeast. So the first thing that popped into my head is, like you said, did they sequence this stuff? Is it from the ribosome, right? Because I mean not the ribosome. Is it from the rDNA locus? Because that's what happens in yeast, or is it in other because the reason why the yeast is so unstable is because the rDNA locus is like 1 megabase and it's very repetitive, and you get a lot of recombination taking place there. Is there, and of course, mammalian DNA and human DNA has a lot of repetitive sequences there. Do these CCFs is a more fundamental process taking place at not specifically at rDNA loci, but just repetitive loci in general that are unstable, and that this sort of happens, and I don't know.
I feel like it's an important question. Like at the end of this paper, they make the point that they've got these HDAC inhibitors that affect this pathway, and hey, maybe these are good drugs that you could take for looking at like aging-associated insults. But one thing which isn't really clear to me after having read this paper and glanced at some other papers by the same authors are whether you like actually want these CCFs or not. Like I could make an argument for CCFs good or CCFs bad.
Yeah.
And like, if you downregulate this, okay, did you make things better? It's not clear.
Yeah. If there's a way to suppress, if you're right, exactly. Is it damage that you want to get out of the nucleus, or is it yeah, like you said, it could be good, it could be bad. If you put a blockade there, is it going to lead to other problems in the nucleus, or is this a problem in of itself? I'm not sure, but I do find it very intriguing, and I think, you know, kind of my spidey sense is telling me that there's something fundamental here that is, you know, if you see something similar in yeast and you see something similar in mammalian cells, and there's some kind of funky similarity here, there's something important happening that's maybe very, very related to aging. I mean, fundamentally, it's genomic instability, right? So it's genomic instability, but it's a peculiar type of genomic instability that is, you know, that you have these bits of DNA coming off and being exported out. And that's kind of the overarching theme of yeast and these mammalian cells here. And the fact that this is happening in Cerevisiae and in human cells tells me that this is probably a universal phenomena of sorts, of something very fundamental taking place, that it's not just a peculiar quirk of IMR90 and Cerevisiae strain, you know, whatever, right? That it's probably all over the place. It's just that not many people are looking at this. Like you said, this is one of the few labs that is looking at this in mammalian cells, and, you know, and no doubt, you know, when other players get pulled in, then it'll pull in a whole bunch of other labs. When people say, hey, P53 signaling is also involved, and hey, a whole bunch of other our favorite proteins are involved, then, you know, you're going to suck in a whole bunch of other researchers. But until then, all we can say is very interesting. Anyway, 5,3-BP1. So that is also 5,3-BP1. So one thing that they mention here is that it's not found in gamma H2AX outside, but it's found co-localized with gamma H2AX inside the nucleus, which would make sense if it's involved in the kind of resection process. But once things get out, it's not out, it's in, right? So it's at the location where these double-strand breaks are occurring, but it's not actually found outside. Okay, so I think what they're doing now is, so they're going to be suppressing 5,3-BP1 using short hairpin RNA. And they're also going to be looking at, I think, so what do they do here? Are they looking at co-localization of 5,3-BP1 with Jun kinase? I think they are. And I think they do show that it's co-localizing using an immunoprecipitation, if I'm not mistaken. See, proliferating. So if you ionize radiation, yeah, so you get Jun kinase co-localized with 5,3-BP1 if you have ionizing radiation. So meaning that it's a suggestive that, this Jun kinase signaling is implicated in, the mitochondrial effect in the nucleus and CCF formation. And now they're showing that 5,3-BP1 and Jun kinase are actually physically binding to some extent when you zap them with ionizing radiation. Of course, that's ionizing radiation. Maybe it's cross-linking them. I don't know, but it might behoove to look at other things as well. I don't know if it co-localizes if you add in other insults as well. But with ionizing radiation, it certainly looks like it co-localizes. Okay, so what are we looking at here? So I'm just jumping to figure D here. So again, 5,3-BP1, gamma 2AX, which is found in these blebbed vesicles that has damage. This is your control, proliferating ionizing radiation, and I believe this is going to be your inhibitor of 5,3-BP1 expression. So everything seems to be okay here for proliferating, but ionizing radiation is where you saw 5,3-BP1. So let's see what is here. Sure. So if you let's see, make sure I got this right. SH53BP1. Which is at 3H. Never had to scroll back and forth so much before in a paper. It's just so much, so much. H, C, E, G, H, I. It's between F-I. Let's see, let me just unpack that. or control lentivirus directed expression of 5,3-BP1. So wait, is that overexpression? Did I get that wrong? It's not. Let me scroll through here. I have to do some editing here. Let's see, which figure am I on even? I'm figure 5 now. No, figure 4. Still have 3 more figures to go. 5,3-BP1, 5,3-BP1, knockdown of 5,3-BP1. Okay. So they're knocking down 5,3-BP1. Okay. So, okay, Jun kinase signaling. Jun kinase is implicated with 5,3-BP1. Ionizing radiation. You get some formation of these bleps, so forming here. If you knock down 5,3-BP1, you get much more. And let's see, so somehow 5,3-BP1 is inhibiting the formation of CCFs. So I'm not sure exactly how it's inhibiting it. So Jun kinase I think Jun kinase is then implicated. Right, so then Jun kinase will be inhibiting 5,3-BP1, which would be inhibiting the CCF formation. And gamma and 5,3-BP1 is involved in resection or repair of DNA, right? So I think the implication is if it can't get repaired, it's getting exported out somehow. So that's probably, might be the sort of the bypass route for if the damaged DNA can't get repaired because you're inhibiting 5,3-BP1. It ends up with more of these blebs. So you basically have an increase of these cytoplasmic chromatin factors, and also you get an increase. So if you inhibit 5,3-BP1, you actually get even more of an increase of these senescence-associated secretory phenotype, because obviously, or these factors that are involved in that, for inflammatory factors, because you're getting more of these CCFs, right? So I guess that's the bottom line here, is that 5,3-BP1 is upstream of CCFs, and it basically holds them in check. And as they say here, it's the first sentence, 5,3-BP1 controls formation of CCFs in the nucleus, right? So it negatively regulates them, and if you get rid of them, then you get basically you get more. You get more of these CCFs. So they've just been putting in the pieces of the puzzle here, where you basically have, check my notes here, bad mitochondria somehow signaling, and I don't think at the end of the paper they quite know exactly how the signals are getting from the mitochondria, the bad mitochondria, to the nucleus. They sort of say, okay, mitochondria are implicated. If you get rid of them, the signaling stuff goes away. If they're there and they're defective, all this stuff happens in the nucleus, culminating in basically an inhibition of 5,3-BP1 and a release of CCFs and all this stuff that happens downstream. I don't think they make that, they're still working on the pathway of signaling between the mitochondria and the nucleus. Like, I'm not sure exactly what is, so yeah, it could just be oxidative stress and then components thereof, or it could be other factors from the mitochondria as well that enter the nucleus. I'm not sure. That triggers this whole response. So I think they kind of leave that still unknown at the end. Okay, so let's go on to figure 5. And now we're doing HDAC inhibitors. Okay, so this is where we kind of get into the interesting stuff, which is, okay, so can you stop this with something that's potentially could be used as a treatment? And they use histone deacetylase inhibitors, which basically would lead to the upregulation of certain genes. And they show that they do get an upregulation. So they showed earlier that you had a downregulation of genes that were that were mitochondrial genes that were in the oxidative, the OXPHOS, for oxidative phosphorylation in mitochondria to generate ATP. Those genes were downregulated when you had bad mitochondria. And later they speculate maybe you have an upregulation of those genes. But anyway, having an HDAC inhibitor, which causes an upregulation of a lot of genes. So they use trichostatin A as an HDAC inhibitor, shows that it improves mitochondrial function and suppresses formation of CCFs, which is your cytoplasmic chromatin fragments, and suppresses expression of SASP, which, is all good. So here is their kind of treatment. This is, I think, still IMR90 cells. So trichostatin A is given beforehand, ionizing radiation, then 10 days later you harvest the cells and you see what takes place. This is just a control. So basically you show acetylation of histone 4K16. So the acetylation status goes up, TSA levels go up, trichostatin A. What they show though is, again, this is kind of uncoupling certain things, is that so earlier they said that some of this mitochondrial, these defects don't seem to affect, I believe, some of these pathways that they're looking at here don't seem to affect the cell cycle, and trichostatin A, if you add that back in, if you add that to these cells through EDU labeling, so basically, I think that's like similar to bromodeoxyuridine labeling. So if DNA is replicating, you have this incorporated, and you can measure the level of incorporation, and the cells are replicating. If you add in TSA, so ionizing radiation proliferation goes down, they get senescent, but you don't get them coming back from, you don't get them re-replicating again. So they're not dividing basically after your ionization blast, even if you add in TSA. But if you add in TSA, the other problems seem to get ameliorated. So the expression of nuclear encoded mitochondrial genes, and no doubt in the materials and methods, and supplementally you can unpack which genes they are, go up with treatment with TSA. So here are some of the genes they look at. Messenger RNA levels go up. MMP, I gotta remind myself, what the heck is MMP? Mitochondrial membrane potential, okay. So mitochondrial membrane potential actually is, well, not quite improved as back to normal, but it is, they say, significantly improved. Looks roughly twofold increase over when you treat them with trichostatin A. The reactive oxygen species go down. Levels of superoxide dismutase also concomitantly go down. And the CCFs go down using this, this assay that they have here when they treat with trichostatin A and various SASP, factors associated with, well, not SASP itself, but the proteins involved, the messenger RNAs, at least for those proteins that are involved in inflammation, go down as well with TSA. And they look at a number of other genes as well that kind of revert back to normal. So I'm going to stop sharing here. 2 more figures to go. So trichostatin A seems to reverse a bunch of things, not everything. So it doesn't reverse your cell cycle halt, but it seems to ameliorate these other problems that are associated, that they implicate with retrograde signaling from bad mitochondria, which is CCF and various inflammatory markers as well. So inflammatory markers, CCF, get squelched by TCA treatment, by an HDAC inhibitor. Gene expression of various mitochondrial genes goes back up. Cell cycle is still halted, right? So that seems to be kind of separate issue that's not involved in this. That's not part of mitochondrial signaling, I believe, in this series of experiments that they've done with IMR90. Okay, I'm going to take a sip of coffee here. So that's where we're at right now, and I think we're getting close to the in vivo stuff that they're going to do with mice. So let me go back to paper. And they're not quite there yet. One more figure. So effects of HDAC inhibitors on other pathways. And they tried basically, I'm going to just, maybe somebody here can comment here, but basically the bottom line is that the HDAC inhibitors, I believe that the pathways they looked at, I think P38, it was one of them. The other pathways that they looked at here were not significantly affected by TSA. So they didn't look at a whole bunch of pathways, but they looked at, I believe, let's see, phospho P38, which pathway is that? Not so MAP kinase. So TSA did not inhibit P38 MAP kinase, and the mTOR pathway as judged by phosphorylation of P38 MAP kinase. So basically, other pathways were, at least the ones they looked at outside of their favorite pathway here, didn't seem to be affected by TSA. But, you know, TSA is a HDAC inhibitor. There's probably a bunch of other things that are affected that they, you know, maybe not looked at. Okay, so that kind of leaves the main figure, which I believe is, well, that's not the main figure, but it's in mice. So this is our in vivo. So figure 7 is HDAC inhibitors suppress inflammation in vivo. So there's a bunch of experiments. So this is a mouse model where they basically give them an overdose of acetaminophen. So basically these poor mice get liver toxicity by given too much Tylenol, I think. They call it APAP, and I'm not sure what APAP stands for. APAP administration. They just say, yeah, I'm not sure what it just acetaminophen. It just seems like a weird acronym for acetaminophen. So APAP overdose. So toxicity of, you get a reactive metabolite and causes mitochondrial oxidative stress, activation of Jun kinase, and this is earlier paper from 2017. So that's the earlier stuff that they show. So liver toxicity. And they showed that they could, if they if they give, I think they use a different, I don't think they use TSA, they use SAHA, S-A-H-A, which is a different HDAC inhibitor. Here we use TSA to complement the previous in vivo experiments with SAHA. What is SAHA? What does it exactly do? What is that acronym?
That one's also known as Varinistat. It's one of the HDAC inhibitors used in psychiatry.
In psychiatry?
I also don't remember what it stands for.
They use an HDAC inhibitor in psychiatry? Interesting. It sounds like interesting. So I guess they're using it for like epigenetic modification of various neurons as an it's used in psychiatry. That's interesting.
Actually, I'm sorry, I might be mistaking. Valproic acid used in psychiatry, and I think it's also an HDAC. But Varinistat. So Varinistat is FDA approved. I think I might be premature about what it's indicated for.
I think you're right. It is used in medicine. I think I'm not sure exactly where. Okay, so they use and I'm not sure what the overall I don't know how much overlap with TSA and SAHA there are. I don't exactly know what the Venn diagram is for those two HDAC inhibitors and how much similarity there is, but they switch gears here. So they go from IMR90 cells to mice and go from TSA to SAHA, which is a different, so C57 black 6 mice, either vehicle HPO beta CD, I don't know what that is, but it's some solvent for SAHA, I guess. Guess DMSO just doesn't cut it for this stuff. And they give it to the mice drinking water. So, And after they basically, so they also switch gears. So instead of using ionizing radiation or something that, like that, they use acetaminophen overdose. So they're looking at liver toxicity. So it's a little bit of apples and oranges comparison, right? So here, some similarity in that there are HDAC inhibitors and there is oxidative stress that's mitochondrial related because of acetaminophen, but different vehicles are used, different compounds that kind of replicate things that were done in vitro in the previous figures. So they show that you have, I think, let's see, so ionizing radiation, and then they look at acetylation status of H3K9, H4K16, different histones. Definitely, it looks like these might be from different mice, I think, samples. And I think you could see here that, the acetylation status goes up for this histone. I'm not sure for H4K16. I have to look and figure, but it does go up for this one. So anyway, controls to show that it's actually acting as a deacetylase. So you get acetylation levels go up at these histones. And what they look at here is gamma H2AX. And I'm not sure an IL-1 alpha, which is a marker for inflammation. Gamma H2AX, I believe this is going to be nuclear. So these look like, what are we looking at here? Blue is blue dapping? No, let me go to the figure here. Make sure I know what this is. So this is immunostaining. So what do we have here? Livers were stained for gamma H2AX and IL-1 alpha and quantified immunohistochemistry images. I don't know what the blue stain is. I guess it's.
I think it's probably just eosin. Like this is
yeah,
an IHC. So it's probably the antibody's brown, and then it's just the usual counter stands.
Oh, right. So it's probably like eosin hematoxylin.
Yeah.
Okay, so it's kind of hard to say that where this gamma H2AX. I guess it's in the nucleus. So I mean that's so you can't really tell if it's blebbed stuff here. I guess that's where I'm trying to go for. I can't tell if this is CCFs or not. I mean, I guess it's just gamma H2AX because of damage, right? So this is so if they so Saha is so interestingly here, yeah. So if they add, so are these cells hit with ionizing radiation, or is this basically just, so here we have, so they say it's not significant here. So, oh yeah, ionizing radiation. So when they hit the cells, yeah, IR, yeah. Too many letters, it's all scrunched together. Saha. It's like, I feel like it's slowly becoming dyslexic. What is Saha? So ionizing radiation treatment, I mean, they say it's non-significant. I guess it's not, but I mean, my eyes can see a difference, but I guess math can't see a difference here. So you do have a significant decrease in after ionizing radiation, and so treatment with the Saha seems to suppress expression of IL-1 alpha. I mean, you could see here, it's pretty clear. I don't know, it kind of looks like it's a difference here. They say it's not significant, but it looks like you're definitely getting less gamma H2AX.
If you zoom in, the right panel has a lot of positive nuclei. They're just really small nuclei.
Oh, I see. So yeah, so maybe they should go for a volume thing as well. I know that was an issue in a different paper where they basically said, okay, you got the same amount of speckles, but there's bigger ones in the left versus the right. So maybe those have the same amount of cells, but the proportion of cells that have more damage goes down. If that makes sense. I'm not sure if they looked at it from that perspective, but they did at least they showed it and they published it, and I could see that. So maybe there is something taking place there with regards to less overall damage within cells, although basically, the same amount of cells have gamma H2AX. So anyway, so I think that's, I think that can't be just quickly discounted. There's probably something happening there, even though it's not significant if you look at the overall number. So then what they do is look at, so that's with, okay, so they do ionizing radiation. They probably just did APAP. So they do APAP as well, and ionizing radiation. So for APAP, acetaminophen, they look at the same thing, and then they add in TSA, and they get a decrease. And in this case, what they're looking at, so they're looking at now at CCF. I don't know why they're not looking at CCF here, but they're looking at CCF, which is cytoplasmic chromatin fragments, and I'm not sure. Oh, so they could zoom in here, and this now gives you, so that's the arrow that's pointing at your bleb. Well, it's a real pain to actually do that via microscopy, right? I mean, geez. They had that automated. So they're able to measure able to measure the cytoplasmic chromatin fragments, and the levels in red, you can see APAP TSA. They basically at the same time give trichostatin A, and the levels go down for also IL-1 alpha as well, inflammatory marker. So it seems that, when you had this treatment, which they've said in previous studies has shown to stress mitochondria in the liver, you do get less of these CCFs. I don't know if they correlate, there's a lot of toxicity happening here, and there's other issues, that lead to basically death of liver, from acetaminophen. I don't know if CCFs are implicated in that. I'm not sure in the death of liver cells with acetaminophen toxicity. Just a thought there. And now there's another immunohistochemistry slide here. What are we looking at here? They're looking at gamma H2AX. I'm not sure what figure F is supposed to, what's the new information there versus what I just saw from E and well, D was ionizing radiation, but E and F. So this is, maybe that's versus, I mean, we looked at gamma H2AX and E. It's just a different, I guess it's just the number of positive cells. So in this case, it goes down. So just the actual number of gamma H2AX positive cells does now significantly go down versus less so significantly with ionizing radiation. Okay. So what they do here, without going over through this, is they look at gene expression profiles of basically, DMSO, TSA controls, and then after APAP treatment, so you got a big change there, this big red bar of basically things getting, regulation getting really messed up, and co-administration with trichostatin A or low doses, which brings levels of various genes that are involved in, I believe, in genes in mitochondria, as well as getting rid of oxidative stress. And they look at some specific ones here: GSTA, HMOX1, SRXN1, and QO1 tend to go back down to normal. So I think there's a lot of, interesting potential in vivo that they could still do, with mice, to basically show that, the signaling pathways that they implicated in IMR90 are also in effect in mice as well. I don't know what else, so I don't know if, clearly also acetaminophen overdose. This is getting a little bit off topic from aging, but acetaminophen overdoses are a huge problem with liver toxicity. I don't know if this then suggests that, you know, certain HDAC inhibitors might have a therapeutic potential, but, you know, in this case here, they co-administered it. So, you know, it's not like they gave it an hour after or a day after when the damage was already done. They gave it at the same time. So it was already there, present, and it seemed to ameliorate that. As far as the mice themselves are concerned, I don't know. I mean, there's no histology there with the liver, and I don't know if the mice, what their survival was after this, right? Are the mice okay? Is their liver regenerated? Is there, you know, I mean, you have these, it seems that there's good things happening, that you basically, that the administration of HDAC inhibitors, which is implicated in the formation, HDAC's formation of CCFs, it basically, but whether or not the mice themselves, actually, I'm not sure if they, because they sacrificed these mice and they did basically, immunostaining on the livers. I'm not sure if you would assume that the mice were better, right? Clinically speaking. But I'm not 100% sure. So, clearly there's a lot of follow-up experiments that would, need to be done. TSA is pretty strong, trichostatin A. I'm not sure. SAHA, I think, is also a pretty strong HDAC inhibitor. I'm not sure if they, they did say that they used lower dosages here that they normally would give. So I'm not sure if those themselves would be a therapeutic compound to give, or if they mentioned at the end of the paper that they're looking at different HDAC inhibitors that they can screen that would have a better toxicity profile. So that's where we're at here. So I think, fundamentally, I'm intrigued by the concept of CCFs as well as maybe kind of a universal phenomena, perhaps, in many other cells. I think it probably exists in many species, and it's, I think these pathways are very conserved. It's just like you mentioned, Michael, there's not many labs that are looking at this. It's kind of niche, but, telomeres were niche many decades ago, right? And of course, everybody knows about telomeres now. Telomeres are all over the place, but it was just seen as some kind of weird thing that protozoans have, right? Like, well, it was speculated, it was theorized, but I mean, the mechanisms for telomerase and extending them, right, was first looked at, was identified in Tetrahymena, and then later, Cerevisiae, and then alt mechanisms were also seen as something weird that was happening in Cerevisiae, but now it's like everything does that, right? So it might be the case here with this, this export of damaged DNA that ties into, retrograde signaling from mitochondria, and this might be an interesting phenomena to look at. And I don't know if the early papers that came out of the Guarani lab, if they implicated mitochondrial signaling with ERC formation. I don't think so. I think they looked at ERCs, they looked at ribosomal DNA instability, but I don't know if they actually made that connection there. Maybe I should dust off those old papers and see. Maybe I could. Maybe there's a paper yet to be written from those papers that I could read and say, hey, let's take a look at the mitochondria. We have a publication there. Really cool. So maybe somebody who's listening to this is in a yeast lab and is like, let me do that right now. And I've given you the glory, so I'll run with it. So yeah, I think there's a lot of cool stuff that is there. I think that I like papers like this. I like that because there's a lot of missing pieces still here, but it's very tantalizing. It's like seeing a jigsaw puzzle that's not complete, but you can sort of make out and guess as to what the missing pieces are, and that there might be a larger tapestry here that includes most species on planet Earth when it comes to this, what's happening here, and it's not just some sort of a niche phenomena with CCFs. Anyway, that's my 2 cents on this. It's a little bit more of a deep dive paper than we usually do. This one kind of, by its very nature, sprawls across several different hallmarks of aging. That was the point that the authors made in the abstract. So, you know, if I had one critique, it's just like, I would like to have one illustration that ties all of these figures together that sort of centralizes it. So we can refer to it, and otherwise I have to keep this model in my head, and as to you know, how everything is interacting here. But otherwise, I guess I have no further comments. I don't know if anybody else has any more comments.
They're all just blown away, Oliver.
Are they? Yes, maybe. You know, there's a lot there. There's a lot there. I mean, you know, mitochondria, nucleus. So, you know, from mitochondria to the nucleus, and then back out of the nucleus again, and then you have a SASP, and then round and round it goes, right? And that SASP probably affects other, you know, cells, which might affect their bad mitochondria, which might affect their nucleus and cause more. Might be a chain reaction there. So it'd be interesting to see if I don't know. Maybe this was in an earlier paper. Do the SASP factors does inflammaging promote CCFs in bystander cells?
I don't know, but it makes healthy cells go senescent.
Yeah, and is part of that senescence CCF, because clearly they tried showing here that that you can sort of separate out the cell cycle arrest part of cell senescence from what's happening with these malfunctioning mitochondria, from the increase in the reactive oxygen species and the CCFs, kind of a different sort of orthologous state of haywireness that's taking place here. That's side by side.
How do we fix it then, Oliver? That's probably the salient question.
HDAC inhibitors.
I don't think that's really the solution.
No.
You know what I'm going to say? Come on. We're not getting to the root of the problem. What's the root of the problem?
Root of the problem? Well, life. I mean, how?
Yeah, well, that's a little bit too vague. What I'm saying is the problem is
back up, too far.
yeah. You've gone way reverse in there, too far. You've gone around the block. But no, it's mitochondria, isn't it? It's the damaged mitochondria that the problem, and it's becoming more and more apparent that mitochondria are playing such a central role in aging. I mean, obviously they've got their own, but they've actually got two hallmarks. In hallmarks of aging, the mitochondria are implicated in two, right? One's causative and one's like a collateral effect. I can't remember which ones, but it's genomic. Is it genomic instability? Yes, they're primarily mentioned in that, but they're also mentioned later as dysfunction. So, yeah, they're big players in aging. So, fix the mitos. How do we fix the mitos? I mean, there are a few companies working on it. Obviously SENS, the program at SENS comes to mind the most, the SENS program, and they're making good progress. I think there might be some, I'm going to go out on a limb and say, there is possibly some news coming soon, hint hint, but I can't say any more at the moment. But the MitoSense program is progressing well, and I think it's a solid solution.
And just to remind everybody of the MitoSense solution, they're basically shuttling. They're putting in. It's like a coin. It's like intra. maybe I'm trying to be too smart for my own good. It's intraorganellar gene therapy. They're transferring mitochondrial genes into the nucleus. So instead of having your mitochondrial genes in the mitochondria, you're shuffling them all into the nucleus where there would be potentially less damaged by reactive oxygen species. So you would sort of have a backup copy of extra genes in the nucleus that I think there's 13 of them in the mitochondria.
There is, yeah, there were thousands, but they've all migrated over the course of millennia to the nucleus as they've integrated more and more with us. There's only 13. I think they've got 4 of the genes out of 13 now, possibly more, but I can't say anything.
Transorganellar gene therapy.
Yeah, in other words, creating a copy of the mitochondrial gene in the nucleus, which is rather cool. I think I also saw another way. Autophagy. Let's talk about autophagy because we can't talk about mitochondria without talking about autophagy. Another possible route to dealing with mutant or damaged mitochondria is inducing autophagy. If you can do that, obviously, autophagy is the cellular recycling program, which includes organelles. So the lysosomes are going to start nomming on all the bits if all the unwanted or damaged bits. So if you can tell the lysosomes to get nomming, i.e. trigger autophagy, that's also another possible solution to, I wouldn't say solve it, but certainly it could potentially slow down that issue.
Well, there's also the therapeutic mitochondrial transfer idea, right? Where you try to put good mitochondria into cells that don't have them.
Yeah, I mean that's been done. Yeah.
What's that paper? There was a paper years ago. I don't know if there's been much. So there was a phenomena in I want to say metazoans. I don't know if they did this in vitro, but basically cells can transport mitochondria from one cell to another using basically sort of like imagine one cell helping another cell by squirting a good mitochondria into the other cell. I didn't read that paper, but I saw a blog about it on a science website like a bunch of years ago. And I was like, oh wait, that's really interesting. I never heard of this phenomena. And this was a couple of years ago. So it was clearly something that was just discovered not that long ago.
Yeah, there's some evidence mitos do transfer as well themselves.
But yeah, but this was a process, and I can't remember. I'd have to Google it and pull it out. But what you're referring to, Michael, is what, like a laboratory methodology for transport rather than a cellular phenomena?
I don't know the details. I think Cell V is the company that's boosting that whole approach. I never heard of cell-to-cell mitochondrial transfer though. That sounds hard, if for no other reason than mitochondria are really big. How do you move that?
So just Googling here, transcellular mitochondrial transfer. I guess that would be the term.
What about the mitochondria that they found in the bloodstream that
I just pulled this up.
Bubbles?
Yeah, I just pulled this up here. Mitochondria can travel from one cell to another by intercellular structures such as tunneling nanotubes and secreted cellular bodies such as microvesicles. It's a long time ago. 2004, Rustam et al. Described TNTs as a structure that enables cell-to-cell interaction. But I think this was, I think there's more recent that suggested mitochondria could do this. This is a review paper from.
There is, Oliver. We covered it last year, or I think it was the year before, where they found bubbles, vesicles, or a bit like exosomes, where there were actual mitochondria fully functioning, floating around in the bloodstream, and they weren't even sure whether they were in the middle of a transfer or whether they were possibly playing a signaling role. So that was the question. They didn't even know whether, what that was. So that opens up another question is, what the hell are the mitos doing out there? And are they also doing signaling duty?
Yeah, so this goes back longer than I thought, more like 15 years plus. But yeah, so I'm looking at mitochondrial transfer between cells can rescue aerobic respiration, 2006, PNS, mitochondrial transfer from bone marrow-derived stromal cells to pulmonary alveoli protects against acute lung injury, Nature Medicine, 2012, mitochondrial transfer, yeah, okay, so interesting.
Those damn mitos. I wouldn't be surprised if they were a very large key as to why we age, and I wouldn't be at all surprised to find out that that's the reason or one of them.
I think evolutionarily they're just trying to escape. That's it. That's.
I don't know. No, they thought it was a good idea at the time.
Running for the lifeboats.
Like, it's like, here, it's a passageway. Let's escape. Oh, damn it, we're in an alveoli. We just went from one cell to another.
So hopefully we'll have a little more current next time. But not to say that mitos are not interesting.
Yeah.
Because I think they are, and they're going to become increasingly more important in the field as people actually realize that the power stations of the cell are, like, probably one of the most important parts of the cell. I mean, don't even get me started on NAD synthesis and all that, which some people just think is tinkering with metabolism, and is not really worth fixing. Yes, it is. So, there's a lot to mitochondria.
Yeah, I mean, a couple of things that we already know about, like you mentioned, obviously, clearly, mitochondria is the powerhouse of the cell, right? They use an old meme here. But yeah, signaling itself, it's at the nexus of measuring kind of the energy state of a cell and all the things that are both positive and negative that come out of that. I mean, apoptosis being regulated by mitochondria, right? That's something that's accepted now, and now we have this retrograde signaling into the nucleus, which can form CCF. So there's a lot of signaling that's happening, in mitochondria, and it's an organelle, so it's got a lot of stuff. So there's going to be a lot of complex signaling activities taking place. So as was mentioned earlier, it's interesting that they were able to get some clean data by just ablating the mitochondria completely out of the cell, just whacking them all out, and basically showing that, that CCFs went away. No doubt, they couldn't keep these things around much longer because the cells don't have any more mitochondria. So you've got a limited amount of time to work with these cells. So I'm surprised that the cells still had the capability to. Yeah, I don't know. I have to read the paper a little bit more. Fortunately,
well, they do, don't they? But
they have to supplement with ATP.
The mitochondria is really what allows our cells to be so efficient. But yeah, I mean, originally our cells didn't have mitochondria, but they kind of sucked. So put it this way, without the mitochondria, we can't have the complex cellular functions that we all enjoy today. And if we've got no complexity, I would probably say that that's why intelligent life evolved in the first place, because of the mitochondria, because they can power all those functions. Think of the mitochondria like, I don't know, your latest iPhone, does all kinds of things. You sure you could have an Android, but like me, but you want an iPhone, don't you? Well, they're kind of like a cellular computer, aren't they? Because they're powering all the myriad and complex processes going on with the cell. In other words, if you want a complex cell and therefore a complex life form, you're going to need a big bloody battery. That's your mitochondria.
Yeah. Things get overly complicated when endosymbiosis kicked in. Things were nice and tranquil for many billions of years, and then endosymbiosis had to happen. And we got dinosaurs. So,
If you believe, yeah, well, that's just your opinion. If you believe in that sort of thing.
Well, I think it's
I mean, come on, the Earth is only 5,000 years old, right? Am I right? So
you're gonna get us kicked off of Facebook.
Yeah, I know. We'll fix that in the dub. All right, guys. Oh dear. Right. We'll sort that in the dub. So anyway, I guess we'll see you all next time.
Yeah.
Stay safe.
All right.
And hopefully we'll all get vaccinated soon if we haven't already. I know John, I believe, already has. And with a bit of luck, we'll all be getting a jab soon. All right, guys. And on that slightly worrying note, I might hopefully see some of you next time. Maybe not Oliver.
I have confidence in my immune system. It's very robust, resilient.
Shouldn't be too robust. That's the problem. It's actually your overreaction. That's the problem with the immune system. Wouldn't be surprised if rapamycin in very low dosage could be useful. But that's another topic, guys. And
Yes.
I'm not going down that rabbit hole. So we'll see you all next time. We'll let you know what the topic is, or if you've got an idea, give us drop us a line. Let us know what you want to talk about. You know, it shouldn't always be about us and what Oliver wants to talk about all the time.
Yeah. Give us some papers and ideas, because that's I'd be interested to figure out what interests everybody out here, and let's hear from everybody out there. So and I'd be happy to dive into something that I haven't seen before.
There you go.
Thanks for all your efforts. Thank you very much.
Thanks very much.
Thank you.
And cheers to everybody who supports us, and we will see you next time.
Take care.
Thank you.
Take care.
Hello.